Good morning and welcome to the class. See, in our last class, we started this new chapter which is on the principles of inheritance and variations. I started with telling you about Mendel, how lucky Mendel was in choosing the pea plant, some of the differences that we see in the pea plant. And not only all this, I also discussed about some of the very nice features in Mendel's experimental design. And then we learnt about the first law of dominance, the genetics laws, which Mendel proposed. We called it as the law of dominance and the law of segregation. And both the laws are something that are very important for a single hybrid studies, which we call as the monohybrid cross. We saw that whenever we take a tall plant, cross it with a dwarf plant in homozygous condition, the first filial generation will always express only the dominant trait, and therefore every plant you see there from F1 generation, they're all tall. Whereas if we allow this F1 to have a self-pollination, give rise to seeds, and there is F2 generation, we saw that F2 generation plants, there is the dominant one which is 3 times more than that of the dwarf ones. So based on these observations, we saw that there were two laws proposed. The first law, which is called as the law of dominance. So these are basically laws which tells that if there are a dissimilar pair of factors like one dominant and one recessive in a heterozygous condition, you see that only the one which is dominating character is the one which is expressed, whereas the recessive is not. So that is the dominant law of dominance. And then we also study about the law of segregation. Now, in the case of law of segregation, we saw that though there are two genes which are present together, or the two factors that are present together, we do not see any blending between them, and the characters just get separated in F2 generation without the blend. And that is the reason why we see that all the siblings of F2 generation, you see many of them resemble, two of them, 50% of them resemble just like the grandparents, having homozygous dominant and homozygous recessive genes respectively, and there are two of them which resemble their heterozygous parents. So this is what we have seen in our last class. But now comes one very important question as to, do we really see this happening all the time? Do we see this kind of gene expression, dominant one always expressed, recessive which is not expressed all the time? Now that is where the Mendel's law kind of fails. I would not say fail, I would say that it just shows some deviation, shows some variation for a reason which we will discuss further. I'm sure you would have taken this plant called Mirabilis jalapa. Have you seen plants of Mirabilis jalapa that looks like that spatica flower? Or maybe you can take Antherinum, that snapdragon, or what we call as the dog flower. Now you can take any of these plants and you see and observe the flower color in them. And you notice that if you cross a red colored flowered plant and a white colored flower plant, then apparently the first generation, F1 generation, is not red or white, but it is pink, which is not resembling any of the parent. But then in the second generation, you see the exact 1:2:1 ratio of red, pink and white flowered plants respectively. Now that is something which is very amazing, completely different from that of Mendel's laws. But yes, this has a profound influence, profound explanation on many of the traits that we see. Now this kind of blending can happen, deviating from Mendel's laws, and this is a phenomenon that we call as the incomplete dominance. Okay, now incomplete dominance just started when we started working the same kind of Mendel's experiment with other plants, and then there was a difference. Okay, so for example, this is generally seen in Antirrhinum, snapdragon, Mirabilis jalipa. We can take many of the plants there, and there what was done was they took the P plant generation P1, and for the P1 generation they took the true breeding plants which were giving the red flowers. Now since they are true breeding, we always write it as capital R, which means to say that they have the same trait passed on to the next generation without any variation, and these were crossed with the white flowered plants. These were called crossed with white colored flowers plants, and these are also true breeding, so we would write this as RR. Now let us see what these gametes are. So the gametes here, would be one type of gametes with capital R, the other one with small r, and in the case of F1 generation, you see that there would be capital R and small r. Now capital R, small r is not white, it is not red colored flowers, but all these are pink colored flowers. Right? Now if I take P2 as capital R, small r, pink colored flowers, cross it with the same pink colored flowers, you see that the gametes are of two types, capital R, small r, something like this, and then in the F2 generation, instead of writing that lines here, I would use a nice easier way of putting that in a tabular column. Now this is called as a Punnett square or a checkerboard. Okay, you call it as Punnett square, or you also call it as a checkerboard. Now in the case of a Punnett square or a checkerboard, we will have the female gametes written on one side, then the male gametes written on the other side. Okay, many of them write it even like this. Same thing, making a square into a rectangle, and you write the gametes here, but I prefer it writing this way, or you can use the one which suits you. Okay, so we write it in the checkerboard here. So the gametes here are capital R, small r, and then we have the capital R and then the small r, right? And then I had told you that whenever you are writing gametes, please do encircle them, because that means that they cannot stay as a haploid condition. This has to be combined, so you always write it in a circle. So let us see what this would be. Capital R, capital R, capital R, small r, capital R, small r, and then finally we have small r and small r. Okay, now let us see this. This capital R would be with the red flowers. This would be pink again. This is again pink, and this would be like the grandparent, which is the white. Okay, so you see that this is slightly deviating from Mendel's laws, but yes, the ratio still remains the same. So what exactly do you understand by dominance? Now, in our previous example, we saw that capital T was completely dominant over small t, whereas in this we see capital R is not completely dominant over small r. Okay. Now, why does this actually happen? Now, if you really want to understand why this happens, you should know what is the importance of this particular, give me, factors. Okay. Now, I would like to erase this part of the board, and then let me tell you that, let me say we have capital T and we have a small t, right? Now, what is the function of this? This is a factor. Now that we are talking about the molecular aspect, we can call it even as a gene. Now, we know that there are two genes. Both the genes are somehow linked to give one trait to the organism. In this case, it is tallness. In this case, it is the flower color, right? Now, if this gene gets expressed, okay, when this gene is there and it starts giving RNA, it gives rise to protein, you see that gives out a trait, right? Now, we have in one cell two copies of them. Now, these both of them are involved to give one trait related to the height of the plant. But please do remember that they are not coding for the same protein. This is slightly different and this is slightly different. Now, this one single gene is not present only in one form, but it is present in many forms. Now, such things are what we call as alleles. Alleles are nothing but the alternate forms of the gene. Okay, the alleles here, you see that they are different, whereas in this particular case, you see that they are the same. So they can be identical as in a they can be identical in a homozygous condition. They can be different in a heterozygous condition. It's not just for many traits, it's not just only two types. And again, Mendel was lucky to choose pea plant because there were only two varying characters. Just imagine what would happen if he chose the skin of a human color. There are so many types of skin. I'm sure you just look at, your friend's skin texture, color. You see that there are so many variants. So there are not just two types. There are many types. So they're all the alternate forms of the genes, and they are called as alleles. Right? Now, what is the function of this allele? The allele should get expressed, and when it gets expressed, it undergoes some kind of changes. So, the DNA gets not converted, it makes RNA, and RNA makes protein, and this RNA giving rise to a protein, the protein gives the characteristic trait. Why is your hair black? It is because your hair follicles, your hair cells makes that protein which gives color. Whereas in older people, that protein is not produced. So you see that their hair turns grey. Right? So one particular protein is giving you one trait. That is what it all means. Okay. Now we will take one example. Okay, let us take the example of a gene which codes for an enzyme X. Okay, now I want this enzyme X, and this enzyme X is produced by some kind of gene. We will call that gene alleles as capital G, and we will take it as small g. Let us assume there are two copies of these genes. Okay, now I do not want capital G. I don't want the protein X. Okay, I'm more interested to study about the genotype. But I can't study genotype if I don't know what this enzyme does. Right? I want to study about this X. What is this X? Types of X, form of X functionality, what is its function, and all that. Okay, now suppose there is one substrate and this substrate has to give rise to product. in the presence of, let me say, this X. Okay, now this X performs this function of converting the substrate to the product, right? But where is this X coming from? Please do note that it is coming from the genes, and this gene can be in any form. It can be in any allele. Okay, it can be not a proper enzyme, or many times you see that the enzyme might not work, or sometimes there is no production at all. Now in each of these cases you see that the phenotype is different, right? So if the X is produced, let me say, like in this condition, it is giving rise to good amount of X, so substrate gets converted to product. Suppose there is presence of, let me say, this GG, let me say this G is dominant. Now then this is not expressed at all, so it is a partially functioning enzyme. And let me say, if the condition exists as GG, small g's, you see that there might be no enzyme present. So therefore this slight modification in the gene is something that plays a very important role in converting the substrate to the product. And this product which is formed might be very well giving you a phenotype. Okay, it might be the tallness, it might be the pink color of a flower, white color of a flower, or it might just not do it, does not have any, phenotypic function at all. So that is also possible. Just to give you one you know example here, there are certain genes which induce cancer. Okay, not induce, but yes, they trigger the formation of cancer. Now why does that happen is because there are certain allelic forms of genes, and these allelic forms of the genes will change the enzyme in a very slight manner, that their enzyme activity changes. And when the enzyme activity changes, you see certain reactive oxygen species increase in the body, and that might lead to the death of the organism in total. So what allele we have and how that allele gets expressed is something which is very important to study and understand. Okay, in the case of gene for tallness, let us see, there is one gene for tallness. Can there be a gene for dwarfness also? It is not there. Therefore you see that there is the plant which kind of grows very well, okay, or it is dwarf. Whereas in the case of flowers, you see that the red color flowers are so dominant that even in the next generation all the flowers appear to be red. Whereas in our incomplete dominance, you saw that red produces some amount, okay, and white is actually absence of the pigment that is also being expressed. So the average of that would be pink, and that is what you see in the next generation. So this way you see the way our genes work is not like, yo, yes, this is the way it is. It does not happen that way. Okay, there are multiple ways, multiple factors, and all of them influence the gene expression, right? Anyway, we will be learning more about these alleles, the gene expression studies, and all that in our next chapter. And also we have one unit on understanding about the central dogma that would also kind of link it to what we are doing, right? Okay, so we have learned about the incomplete dominance. Now we need to discuss about the next aspect, which is called as the codominance. Okay, let us talk about one more interesting feature, which is called as the codominance. Now what is codominance? Incomplete dominance is what? Dominant feature which is incomplete. So red is not completely dominant over white, right? Now codominance is what? Now both are dominant, right? It is both dominance. So in the case of codominance, you see that the F1 generation resembles both the parents. Okay, it resembles both the parents. Okay, so how does it resemble? Say, I would like to give you this example of the cattle skin. The cattle skin, it is brown. Let me say there is brown parent and white parent, and if it crosses, you just look at it. Brown hair is also seen, white hair is also seen. Have you noticed that? Yes, that kind of existence of both the genes together is what we call as the codominance. Okay, now this is very commonly seen in the blood group of humans, which is called as the ABO blood grouping. ABO blood grouping in humans. So what happens in ABO blood grouping is that blood group is controlled by one gene which we call as I. Okay, what is the function of this I? This I gene produces something that we call as antibodies. Okay, so suppose we have this red blood cell. Imagine that this is a red blood cell. Now on its surface, you have certain protruding factors. Okay, now these protruding factors are basically small proteins having sugar chains also. Okay, so it is made up of proteins and sugar chain that comes out from the plasma membrane. Now this gene I is involved in making those antibodies, in making those uh, you know structures on the plasma membrane. Okay. Now apparently there are uh, three different alleles of I. Okay. What are alleles? Alternate forms of genes. So I need to write them. So how I write it them is I will write it as IA, then I write it as IB, and then there is a recessive form which is called as small I. Okay. So there are IA, IB and I. Okay. Now, if let me say if there is IA, it produces, hypothetical children, it's not like this, it produces one kind of sugar chain. Okay. And if it is IB, it produces slightly different sugar chain. Whereas if there is I, it does not produce any sugar chain. Okay, so basically there are three different alleles. Okay, and they produce slightly different forms of the sugar, you know, for it to be produced on the plasma membrane. Okay, but now we all know that we human beings are, diploid. Now, because we are diploid, we have a different combination of each one of these genes. Okay, we have different combinations. Suppose I have IA and IA, then you see the blood groups will always have this kind of antibodies present on them, and therefore the blood group is what we call as A. Okay, there is one more combination. If it is IA and I, even then you see that the blood group will be like this. So this is nothing but same, right? So this is also a blood group. Similarly, there can be IB or IB with the small I. Now in such a condition, you see that the blood cell would have an antibody like this, right? But suppose there is one IA and one IB, what do you think would be the structure? Now in this case, it is having a cell structure like this with A and with B together, because we are talking about one phenomenon which is called as codominance. A is also expressed, B is also expressed, and equally expressed. It's not just partially expressing, equally expressed, such that on the red blood cell you have A as well as the B antibody. And sometimes there is also a possibility for getting small I. Now in them there is just one red blood cell, right? Did you get the point? So these are some of the combinations that or combinations of the genes that we might have, and yes, that is also the phenotype that we were talking to. Now if you understand this mechanism, you will also get to know which blood can be used as universal donor, which blood uh, is, you know, universal acceptor, and so on. Okay, yes, but though we talk about A, B, O blood group, uh, frankly speaking, this is not the only one which is used for blood transfusion. This is a major one, but along with that we have something called as the major histocompatibility complex and many more. But yes, that we will discuss in a later part of biology. So I hope you have kind of understood how this blood grouping system works in our body, and this is how the codominance works. Okay, so now you see that this also exists something that we call as the multiple alleles. Okay, sometimes you see it's not just tall and dwarf, white and yellow. You see that A, B and O blood groups. So there are so many types of alleles for many of the genes that we spoke of. I was telling you about the cancer. All the cancerous, all the genes that are involved here in cancer, you see that they are all exhibiting multi-allelic form. Okay, but what exactly are multiple alleles and how many alleles are present? Now that's a huge study which has to be done. In fact, that is one reason why Human Genome Project is kind of still to be understood well, because for every gene we have so many types of alleles. Okay, uh, just wanted to tell you about one interesting feature here, and that is, you know, we always think that one gene will have one effect, right? But it is not so. Many times we see that it produces more than one effect. Okay, uh, there is starch synthesis in the pea plant. Okay, and that is controlled by one gene which is called as B. Okay, B. Starch gene is starch production is controlled by by a gene which is called as B gene. But you see that this is not, this exhibits multiple allelism, or and you see capital B and small b. It's not multiple, but yeah, it is exhibiting two alleles. Okay, now you see that if there is capital B and capital B, now such plants are homozygotic, right? And you see the large starch grains are produced, right? Whereas if it is small b and small b, then you see that they are much uh, smaller starch grains. Okay, this BB seeds are always round and this is always wrinkled, right? So you see that B is considered to be the dominant allele, right? This is what happens. But if by chance we have capital B and small b, okay, if it is BB, you see uh, it does not show a complete dominant character. They show some kind of incomplete dominance. Okay, so this is more or less like round but not completely wrinkled, and you also see the starch grain size is the phenotype. So it's like starch synthesis is happening, controlled by two genes, and you see it is not just helping in starch synthesis, it helps in starch synthesis, starch storage, and also the size, the texture of the seeds. So all this also is something that is very important, all carried out by one single gene, which is called as the B gene, right? So just to conclude about dominance, we say dominant, it's not an autonomous feature of a gene. Okay, it depends as much on the gene product, the type of the phenotype, the particular phenotype that we choose to see, and what are the different genes that get influenced because of this. Okay, so this is what is about the law of uh, dominance, law of segregation, incomplete dominance, and the codominance. Any doubts with this? Now, if you do not have any doubt, I would just ask you one question as a homework, and I would like you people to do that so that we can discuss about that in our live class. So, a child has a O blood group. Just imagine the child has a O blood group, and we know the blood groups of father and the mother, which is A and B. Okay. Now, in such a scenario, what do you think would be the phenotype, the genotype, the gametes of the parents? And if by chance they have one more child, what do you think would be their possible genotypes? Okay, so child is O. I want you to work out with the genotypes of the parents and also the possible genotypes of the offspring. Okay, so good luck. See you later. In our live class. By the time, I wish you do this particular problem that's told to you, and we will have class in our next class. See you there.